Welcome to today's video. In this video session, we are going to discuss about the Krebs cycle, which is also called as tricarboxylic acid cycle or citric acid cycle. Why is it so called? It is called as Krebs cycle based on after the discoverer. This was discovered by Krebs and also Woods et al. The other scientists, but the major work was done by chemistry work was done by Krebs, Hans Krebs. Hence, it is called as Krebs cycle. And the first stable product to be formed is the citric acid, or the first product to be formed in the cyclic reaction is citric acid. Hence, it is also called as citric acid cycle. And as we all know, citric acid is a carboxylic acid. It contains three carbon compounds, C3 compound. Hence, it is called as tricarboxylic acid. Tri means 3, so it is a carboxylic group. So tricarboxylic acid cycle or TCA cycle it is called as. So this is the second step that occurs in the respiration of plants or animals. The respiration is almost same. So after glycolysis, we know two molecules of pyruvic acid are formed. Then it undergoes Krebs cycle. Then the last product is the terminal oxidation and oxidative phosphorylation. So for those people who haven't watched the introductory part of respiration, glycolysis, I advise you to please go watch those videos, then come back to these videos. Then you will understand very well. And here even I have a mnemonic for this code for this, which I'll be saying at the last. So once you're all familiar with the names, I'll be saying you the code. So it's very easy to remember. So just go through those videos, then come back to these videos. So as I said you, this is called as mitochondrial respiration also, because it occurs in mitochondria, whereas the glycolysis was called as cytoplasmic respiration because it occurred in cytoplasm. This occurs in the mitochondrial matrix, and it is called as mitochondrial respiration. Here you have to remember the names and the enzymes. So from the first video I am saying you that it is very easy to remember this cycle if you remember the enzymes or if you remember the action of enzyme if you do. So if you take any enzyme, it will be having two parts according to the International Union for Nomenclature of Enzyme. The first part will be the substrate name, on what substrate it is acting, and the second part will be the function that it is doing. For example, if I am an enzyme and if I am acting on fumaric acid and if I am oxidizing the fumaric acid, then my name will be fumarate oxidase. If I am dehydrogenating, that is if I am removing the hydrogen, then my name will be fumaric dehydrogenase. If I am removing carbon dioxide, that is decarboxylation, my name will be fumaric decarboxylase. So you have to remember this, all the simple tricks that you have to remember, then this will make the entire cycle easy for you. So let us start. So the pyruvic acid, that is two molecules of pyruvic acid is released due to the breakdown of glucose or carbohydrates or proteins, that is the product of glycolysis. End product of glycolysis is 2 molecules of pyruvic acid and 8 molecules of ATP to be remembered. I'll discuss the ATPs and all later. So from the 2 molecules of pyruvic acid, acetyl coenzyme A has to be synthesized. So for the production of acetyl coenzyme A, there are 2 steps. Either it may undergo oxidative decarboxylation. The name itself says oxidation is occurring, decarboxylation is occurring, removal of carbon dioxide. Or it may also, this acetyl coenzyme may also come from beta oxidation of fatty acids, which I have dealt in another video. So when you do the beta oxidation pathway study of fatty acids, the acetyl coenzyme A is the final product. So acetyl coenzyme A is a raw product for Krebs cycle, or it is the starting product we can say. It is a raw material without which the Krebs cycle won't occur. So the source for this acetyl coenzyme A for starting Krebs cycle is obtained from 2 methods: oxidative decarboxylation of pyruvic acid, which is the end product of glycolysis, or it may come from beta oxidation. So now we have got acetyl coenzyme A. So from pyruvic acid, in the enzyme pyruvic acid dehydrogenase decarboxylase. So if the names are not clear, just listen to me properly and make a note in your book. Pyruvic acid. So pyruvic acid, it combines with enzyme called as pyruvic acid dehydrogenase. I said you right. Pyruvic acid dehydrogenase. The first part is a substrate. So it acts on pyruvic acid. What does it do? It removes hydrogen and it removes carbon dioxide. So the name is pyruvic acid dehydrogenase or decarboxylase. So it acts on pyruvic acid and it removes carbon dioxide. As I say, carbon dioxide is coming out. So carbon dioxide is evolved into the atmosphere. When we respire, we give out carbon dioxide. From where does this carbon dioxide come? So it comes from this path only. So and it takes in coenzyme A. So this pyruvic acid, in presence of coenzyme A and enzyme pyruvic acid dehydrogenase, it gives rise to acetyl coenzyme A. This acetyl coenzyme A is a C2 molecule. This is C3. Pyruvic acid is a C3. That means it has three carbon compounds. One carbon is gone out in the form of carbon dioxide. So how much carbon dioxide will be left? I mean, how much carbon will be left? Two carbon will be left. That is acetyl coenzyme A with two carbon atom. It combines with oxaloacetic acid. Oxaloacetic acid is a four carbon compound. So when four carbon compound and two carbon compound combines, you'll get a six carbon compound, right? That is citric acid. 4 plus 2, it is citric acid. This citric acid, and then how does it come? In the presence of citric acid synthetase. The presence of citric acid synthetase, you will get citric acid. And this citric acid is a it contains six carbon compound. It's first stable compound, and it is called a citric acid cycle. And majority of the compounds will have only three carbons or three carboxylic group. Hence the name is tricarboxylic acid cycle. Three carboxylic group will be present. So the citric acid is the first formed product with the enzyme citric acid synthetase, and coenzyme A will be given out. Whatever it takes, it will given off. So this is another trick. So when you put CO, that is coenzyme A, we put arrow mark inwards. The next step you have to put arrow mark outwards. So these are very important. These names you can remember within a minute with the code I am giving you. So the citric acid next it converts into cis-aconitic acid in the presence of aconitase enzyme, and water is liberated out. Now the cis-aconitic acid is then broken down into, or it gives rise to isocitric acid by taking a molecule of water in the presence of an enzyme aconitase. Isocitric acid is then converted into oxalosuccinic acid in the presence of isocitric dehydrogenase. Dehydrogenation is happening. Isocitric is the substrate. So whenever this dehydrogenation taking place, this hydrogen molecule becomes NADP, takes this hydrogen molecule to become NADPH2. Here also the first dehydrogenation occurs, so hydrogen is liberated out. This hydrogen is taken by NADP and it becomes NADPH2. Similarly, here also dehydrogenation is occurring. NADP takes this hydrogen molecule and becomes NADPH2. So we formed oxalosuccinic acid. This oxalosuccinic acid forms alpha ketoglutaric acid in the presence of oxalosuccinic acid decarboxylase. So carbon dioxide is given out. This also when these all are six carbon compounds, when carbon dioxide is given out, so one carbon is lost, so it becomes 5 carbon compound. So alpha ketoglutaric acid is a 5 carbon compound. Now this alpha ketoglutaric acid undergoes again decarboxylation. So the enzyme is alpha ketoglutaric acid decarboxylase. Substrate is alpha ketoglutaric acid. It undergoes decarboxylation. Hence the enzyme name is alpha ketoglutaric acid decarboxylase. Many people say that it's difficult to remember the enzymes. So if you just remember the names and what is happening, whether dehydrogenation or decarboxylation or oxidation, enzyme can be written very easily. So in decarboxylation, one more carbon dioxide goes off. So there were 5 carbon, one more carbon went, it became C4 carbon. So the succinyl coenzyme A is formed and dehydrogenation also occurs over here. So as a result, NADP is converted to NADPH2. So succinyl acid is formed. Succinyl coenzyme is formed. Succinyl coenzyme, again it forms succinic acid, and now what happens? GDP gets converted into GTP. There is one phosphate group over there which gives up, it takes one more phosphate group from succinyl coenzyme A and it becomes succinic acid. So GDP gets converted into GTP. Succinic acid in the presence of succinic acid dehydrogenase. Dehydrogenase means the hydrogen is removed and this hydrogen is now accepted by FAD. FAD becomes FADH2 now. So succinic acid becomes fumaric acid. This fumaric acid in the presence of enzyme fumarase, it takes water molecules and it becomes malic acid. Malic acid in the presence of enzyme malic acid dehydrogenase, it undergoes dehydrogenation. That hydrogen is accepted by NADP to form NADPH2 and malic acid becomes oxaloacetic acid. Oxaloacetic acid, again it combines with another molecule of acetyl coenzyme A to form citric acid, and same steps occur and the cyclic reaction, so it keeps on occurring. So I hope this much is clear. So you have to remember now what is the significance of this Krebs cycle. This Krebs cycle produces enormous amount of energy-rich molecules such as ATP, NADPH2, GTP, FADH2 it produces. So let us calculate the energetics of Krebs cycle. What is the energy that is produced? So for that we should know how much NADP is produced. So one NADP is produced over here, second NADP is produced here, third NADPH2 and fourth NADPH2. So you have 4 NADPH2 molecule is produced. So you have 4 NADPH2 molecule. One NADPH2, if it undergoes terminal oxidation and oxidative phosphorylation, it gives 3 ATP molecule. So if 4 NADPH2 undergoes oxidation and of oxidative phosphorylation, 4 into 3, 12 ATPs will be formed. I hope you understood this. 1 NADPH2 gives 3 ATP. Then how much does 4 NADPH2 gives? 4 into 3 multiplication, 12 ATP. So that is of NADPH2. What is the next energy rich molecule that is obtained? GTP. So this GTP that is obtained, it is similar that as ATP. It does not undergo any oxidative phosphorylation and all. So 1 GTP is equals to 1 ATP. So this is nothing but equals to 1 ATP. It is equals to 1 ATP. Next to this FADH2. This FADH2 also undergoes oxidative phosphorylation. 1 FADH2 gives 2 ATP. So only 1 FADH2 is produced in the entire cycle. So you get 2 ATP molecules. So 12 ATP from NADPH2, 1 ATP from GTP and 2 ATP from FADH2. So if you see 12 plus 2 plus 1, 15 ATP molecules is produced from one molecule of pyruvic acid. But in the glycolysis, how much pyruvic acid we got? We got 2 molecules of pyruvic acid. So one molecule gives rise to 15 ATP, means 2 molecules will give rise to 30, 15 into 2. So 2 molecules will give rise to 30 ATP. So this 30 ATP is the significant product of this pyruvic acid, and glycolysis produces 8 ATP molecules. So in the entire respiration, you get 38 molecules of ATP in aerobic respiration, and this happens only in the presence of oxygen. Only in the presence of oxygen. This is aerobic respiration, does not take place in anaerobic respiration. So this is about the energetics, bioenergetics. Energetics involve significance of Krebs cycle. Now coming part to the last part, how to remember this. So if you just remember one code that I give you, you can write the entire Krebs cycle. Pack square IOK S square FMO. So I will write it here. In this, you can't remember the enzymes. This is just the substrate molecules you will remember. You have to write the enzymes. By doing so, as I mentioned in the trick, when you know what enzyme, what substrate, what is it function, you can write the enzyme. Pack square, PAC square, pack square, pack square. P for pyruvic acid, A for acetyl coenzyme, C square means C and C. First C stands for citric acid, the second C stands for cis-aconitic acid. Pack square. Next is IOK. IOK. I for isocitric acid, O for oxalosuccinic acid, K for alpha ketoglutarate acid. Keto starts from K, right? So alpha ketoglutarate acid. S square. S into S. Succinyl coenzyme A and succinic acid stands for S square. X for first S is succinyl coenzyme, second S is succinic acid. Next is FMO. FMO. Fumaric acid, malic acid, oxaloacetic acid. So this is as simple as this. If you just remember this much, the entire Krebs cycle becomes easy. So pack square IOK S square FMO. So pack square IOK S square FMO. So within a minute, I can say the entire substrate: the pyruvic acid, acetic acid, citric acid, cis-aconitic acid, isocitric acid, oxalosuccinic acid, alpha-ketoglutarate acid, succinyl coenzyme A. One more S is there: succinic acid, fumaric acid, malic acid, oxaloacetic acid. So with just this, you can write the entire Krebs cycle. So I think I made this entire concept clear for you and easy for you. Just remember this code: pack square, IOK, S square, FMO. Keep repeating on this and keep deriving this. Using this, derive those things. Read it front, read it back, so that when you're well versed with this code, the entire Krebs cycle will be easy for you. So thank you for watching this video. Please do like my channel and subscribe to my channel for further good videos on biology and all. And please post your comments and suggestions. I'll try to improve myself. And please share this video with all your friends and groups. Thank you. Please do like and subscribe to my channel.